Hello, everybody. This is the recording of Chapter 5, Part 1. Why you don't see a live lecture about this part will be explained in a message in your class show. So if you're watching this video, you should already have the explanation. The second part of Chapter 5 will be covered either through a live lecture or maybe through another recording. The message in your flash should be explaining that too. So without any further ado, let's come to Chapter 5. In the lab, you have seen a good deal of detail about the integumentary system. We will go a little bit greater detail here in the chapter. So in this first recording, first part of Chapter 5, we will be covering general properties of the integumentary system and the epidermis. So let me go into screen sharing. I'll be showing you the lecture slides and a few things I found on the internet. So here goes integumentary system, part 1. And I hope you can see this. I'm going to switch to full screen here. I'm going to minimize this guy in the corner. So this is the model of our integumentary system you see in our textbook, maybe in the lab manual too. Later on the lecture slides, you will see our lab model because your mind is either still in or just a few days after the practical mode. So, but you can recognize the general characteristics, two large layers, epidermis and dermis, even the subcutaneous layer or hypodermis. So just like in a lab, we have to state that the integumentary system is made up of the skin plus the accessory structures that you see here in abundance, hair follicle, and so on. So let's take a look at some general characteristics of this integumentary system. So functions coming from skin, and the title should say plus accessory system, plus accessory structures. One is a protective function, physical, chemical, biological barriers. In the lab, we mentioned stratum corneum, and this is just by its thickness, the dry nature, is a tremendous chemical and biological barrier. The best physical barrier is actually the dermis. The physical protective system is the dermis that comes on top of the protective function of the epidermis. In the epidermis, in between the epithelial cells, we see tight cell junctions. We also see desmosomes. Lipids are released from the accessory glands, mostly the sebaceous and the apocrine sweat glands. We'll talk a little bit here today about pigments, namely melanin, a little more than what we talked about in the lab. And we also mentioned Langerhans cells, which is part of the outposts of our immune system and react to invaders that do penetrate the epidermis. In our integumentary system, we do have several elements of a nervous system that detect changes in the environment. In the lab, we covered Meissner corpuscle that detects touch, Pacini corpuscle that detects pressure, but we have individual cells of the nervous system that can detect tickle, heat, cold, pain, and even vibration. So in NP2, we'll expand on these Meissner corpuscle, Pacini corpuscle duo. Here we just state that through our integumentary system, we can detect a good number of changes in our environment, and therefore we can take action to preserve homeostasis. In terms of thermoregulation, in the lab, we mentioned the eccrine sweat gland, which is producing the eccrine sweat. And that water liquid, upon evaporation, takes so much heat energy with it that it lowers body temperature. Another aspect of thermoregulation comes from the blood vessels in the dermis. We didn't mention this in the lab at all. So if you think about the body as a radiator, usually the air temperature is cooler and core body temperature, even cooler than the surface temperature of our skin. So we constantly radiate heat into our environment. Now, if we are overheating, then we can increase the blood flow to the dermis, and this warm liquid will bring more heat to the integumental system, so we can radiate, we can get rid of more unneeded heat in our body. So blood flow to the dermis is another aspect of thermoregulation. If we are falling below core body temperature, if our core body temperature is falling below normal, then the blood flow to the dermis is cut back. This blood flow adjustment is done through the either dilation of blood vessels, called vasodilation, that brings more blood to the dermis, or vasoconstriction that narrows blood vessels, and that will bring less blood to the dermis. I want to say a few words about this mysterious function, blood reservoir. Reservoir tends to mean storing of some kind of liquid, like a lake-like reservoir. Because of the large size of the skin, 10 pounds, it has a large number of blood vessels, and blood flow slows down in our veins. About 70% of the blood volume in your body, as you're listening to this recording, is found in your veins. So when we are conducting a sedentary lifestyle, sitting, maybe sleeping, we don't need a lot of blood to carry oxygen, and so veins dilate and kind of slow down blood flow. That happens in our skin, and this is just by the virtue of its large size. Our skin can serve as the most significant blood reservoir during our sedentary life. Another aspect of integumental system is synthesis of vitamin D. We have to explain this here, that vitamins usually mean chemicals that our body cannot make. We have to consume them in order to maintain homeostasis. Vitamin D, as it turns out, initially was thought to be needed from the outside. Later, biochemists discovered that vitamin D is made in the body. And the secret to making vitamin D in the body is that the first reaction needs to be energized, activated by UV light, ultraviolet light. This can happen only in the skin, because deeper parts of the body, such as liver and kidneys, cannot get any UV light, because all UV light is blocked by the surrounding tissues. So synthesis of vitamin D starts in the skin, and this partially made product will move on to the liver. Another modification or two will be made in the liver, and then vitamin D is activated and completed in the kidneys. And the significance of vitamin D, we'll see this in chapter 6. It is stimulating calcium absorption from the digestive tract. For the function of the skin, we have to mention excretion. This doesn't seem like a whole lot of doing anything. In AP2, we'll talk about the water budgeting of the body, and we'll see that there are a number of automated water losses from our body. One is this the baseline water evaporation through our acrine sweat glands. It adds up to almost to a volume of a plastic water bottle. So this is part of the automated water loss from our body. The salt, carbon dioxide, ammonia, urea is just there for curiosity purposes. We have organ systems that are a lot better able to remove salts, ammonia, and urea, namely the urinary system. When it comes to carbon dioxide, our respiratory system removes a lot more carbon dioxide than acrine sweat gland. So here is the first appearance of this lab model, just to indicate that the basic elements of the integumentary system we discussed in lab. So about 22 square feet, 10 pounds. And we mentioned this in lab. The basic structure of our integumentary system is two main layers, the epidermis and the dermis, and then adding accessory structures to it. So among the organ systems, I think you'll agree by the end of ANP1, definitely ANP2, that this is a fairly simple organ system. And in addition to the epidermis and dermis, we have to mention here the hypodermis or subcutaneous layer. This is a layer rich in adipose and areolar connective tissues. While the dermis is made up of areolar connective tissue immediately under the epidermis in the pupillary region. And we have the dense irregular connective tissue making most of the reticular region of the dermis. And the most superficial large layer to our skin is the epidermis, and this is stratified squamous epithelium by tissue type. So let's take a look at this illustration. In the lab, we saw this very nicely illustrated in one of the microscope slides. So we have seen elements of the dermis on the slide. This is a great illustration here that the dermis, being a connective tissue, is rich in blood vessels. You see the stratum basale here, and stratum spinosum, granulosum, lucidum, and stratum corneum. This is a great illustration of how the stratum basale cells are pushing the daughter cells on top of them in superficial direction. And we mentioned this in the lab too. This is a key to building the stratum spinosum and the rest of the layers. We have here the Langerhans cell. It's named a little more convoluted in this illustration, but this is an element of our immune system, and this cell can attack invaders, and if needed, call the rest of the immune system to the rescue. We also see one sensory structure here. Sometimes this is called a Merkel disc, sometimes tactile disc. This is a light touch receptor. And we'll come back to this in a little bit. We see a melanocyte here in the lab slides. This is invisible, but you can see this dark globules, the melanin made by this cell, and eventually the melanocyte will be exocytosed all throughout the epidermis. So we're going to go layer by layer and look at the characteristics and the mechanism of building the epidermis. See the direction here, superficial deep. We're going to start from a deep orientation, from the deepest cell layer, because this is crucial to build the rest of the epidermis. So here is stratum basale. On this illustration, you see a layer of cuboidal-shaped cells. So these epithelial cells represent stratum basale. We mentioned in the lab that this is the site for constant cell division, so the stem cells of our epidermis are located here. And when you look at the dermis underneath, take a look at these blood vessels. Here's one, two, there's three. And so these blood vessels bringing nutrients, blood sugar, amino acids, oxygen to this location. And those chemicals, once they get out of the blood vessels, they diffuse into the epidermis, moving to a place of high, in the dermis to a place of low, in epidermis. And so the stratum basale cells are catching a rich flow of those nutrients, and this is why the stratum basale cells are able to divide constantly. There's limitless nutrients and oxygen that they can rely on. So even the stratum basale cells, if you want a designated cell name for them, they're called keratinocytes. And so are the stratum spinosum, granulosum lucidum, or corneum cells. So we're looking at hundreds of keratinocytes here in this layer. So the stratum basale cells are the only cells in the epidermis that can divide constantly. And we'll see another comment about this a little later. If you want to replace injured epidermis or even injured skin, let's say a firefighter loses 30% of their epidermis to a fire, then skin grafting must include either snippets or large sheets of replacement tissue that must have stratum basale. Otherwise, the grafted epidermis will not survive. Stratum spinosum cells cannot rejuvenate themselves, cannot divide. So stratum basale cells important in maintaining and also healing an injured epidermis. Because in the stratum basale we see melanocytes, this is where it's best to talk about skin color pigments. So we covered this much in the lab. Melanin is made by melanocytes. Now, how melanin is made, melanocytes use this amino acid tyrosine and stitch together hundreds of these tyrosine molecules to create this dark brown globules we call melanin. Melanin is protecting the stratum basale cells against UV light, and interestingly, when melanocytes are exposed to more UV light, they increase melanin production. If you've ever done tanning on in sunlight, this is the mechanism. You are, during tanning, exposing your melanocytes to UV light, and they respond by making more melanin and therefore making your skin color darker. So talking about skin color, when you look around in a classroom or any place you go in this great country, we are seeing different skin colors, and whether it's a pale skin color or a very dark brown skin color, it's interesting to know that everybody, regardless of the depth or darkness of brown coloration, we all have the same number of melanocytes. So how is it possible that some individuals have very dark skin color, meaning their melanocytes produce a lot of melanin, and there are very light skin-colored individuals, and it kind of makes sense that in those individuals, melanocytes produce very little melanin. So this has to do with the enzyme tyrosinase. So under regular circumstances, in a dark brown colored person, the melanocytes have tyrosinase that's working perfectly, flawlessly, producing a lot of natural melanin. In paler skin-colored individuals, tyrosinase enzyme is slow, maybe because of some mutations, maybe for some other reasons. And so in lighter skin-colored individuals, it's the tyrosinase defect that produces fewer melanin and therefore very little pigmentation. The extreme of the disability of tyrosinase is reflected in a genetic disorder called albinism. So albinism, in albino individuals, there is no active tyrosinase. Sometimes there is no tyrosinase whatsoever, and that is because of the disablement or large change to the gene that encodes this enzyme, tyrosinase. So let me go to an image here and change the screen sharing. And let me go to this. I think this is the screen share you want right now. So here's a person with albinism, and you can see the lack of coloration, pigmentation. The pinkish hues are coming through because of the color of blood in the dermis shining through the epidermis. Albinism is a relatively harmless genetic disorder, although we have to mention that, and you'll see this in AMP2 in greater detail. There is a layer in our eye, right behind the retina, that absorbs stray light, and that absorption needs melanin. So in albino individuals, sometimes there are visual impairment because that absorbing layer is missing and light bounces around inside the eye of an albino individual. Another disorder related to melanin formation is vitiligo. Well, somebody says vitiligo. Well, here's the effect. So this is now, vitiligo is an autoimmune disorder. The immune system of this person mistakenly attacked melanocytes, killed them, and therefore no melanin is produced in those areas. In some other areas, the immune system have not attacked the melanocytes, and the natural skin color is showing. So this is yet another evidence that we can get lack of pigmentation not only by removing tyrosinase enzyme, like in albinism, but by killing melanocytes, which again removes the active tyrosinase, and we come back to a skin area or skin areas without pigmentation. Let me go back to the lecture slides here. It's important to mention also here that, this is not typed out on this slide, we have skin color changes such as cyanosis, appears mostly on the lips. This indicates, this bluish coloration of the lips indicates oxygen deprivation, oxygen shortage. We also have the yellow skin color coming from the disorder of jaundice. And jaundice is a malfunctioning of our liver where this yellow colored pigment is entering into the blood when it's not supposed to. There are a number of reasons for jaundice to develop. Usually it's some kind of damage to the liver, either through hepatitis C, cirrhosis, liver cancer, or maybe just physical trauma to the liver. So those cyanosis and jaundice are unnatural pigments. Melanin is the only natural pigment in our integumentary system. When we move to the stratum spinosum, we'll stop here also for a minute because while the cells no longer divide here, they still hold on to each other, as they will all throughout the entire epidermis. They hold on to each other through desmosomes. So desmosomes are incredibly important for the integrity of the epidermis. So if you want to maintain this barrier function, you better have healthy desmosomes. So I want to show you a genetic disorder where the desmosomes are damaged. This disorder is called epidermolysis bullosa. In this case, the intermediate filament part of the desmosome is not forming well. Those anchoring fibers are short or broken, and the cells are no longer holding onto each other very well. So let me go to another screen share and show you an epidermolysis bullosa. This is not that. Here is our image. So this infant was born with epidermolysis bullosa. You can see that the epidermis is no longer holding together. It is coming apart left and right. And this indicates that the desmosomes are no longer holding into each other, and relatively light trauma to the skin leads to the breaking of the epidermis. Now, underneath this pinkish tissue is the dermis, so there's nothing wrong with the dermis in these genetic conditions. Some epidermolysis bullosa cases are so severe that they are not survivable. With the breaking of the epidermis, we lose that biological barrier function, so bacteria, fungal spores, viruses can attack and get into the body through the dermis. There are milder forms of this disorder. Sometimes epidermolysis bullosa shows up as a light blistering disorder or a weak effect on the integrity of the epidermis. So this is all coming from the effect of desmosomes, mostly in the stratum spinosum. The next layer, as the stratum spinosum layers being pushed away from the stratum basale by the constant cell division in the stratum basale, right? So the stratum spinosum cells are like on an escalator and slowly being moved up. And once they move far away from the dermis, the cells begin to die. This is the story in the stratum granulosum. The cells are darker than the neighboring cells because granules loaded with lipids and protein, keratin-like proteins, are making the cells dark. So this is stratum granulosum. And when this escalator keeps going up, superficially, the cells finally pushed far away from the nutrient source in the dermis, that cells die. So the stratum lucidum, which is coming from thick skin areas of the body, such as palm of the hand, bottom of the feet, stratum lucidum cells are totally dead. And you can see here a little evidence of why this is called lucidum, because it's brighter, lighter in coloration than the neighboring cell layer. And No surprise, stratum corneum is also made up of a layer of dead cells. Now the escalator from the stratum basale has pushed cells even further away from the nutrient source. The stratum corneum cells are still holding onto each other because of those desmosomes. We slowly lose them as we put clothes on, we take a shower, or if we just happen to be rubbing against something. And the stratum corneum is where this tremendous protective barrier function comes. Stratum corneum is nearly impenetrable to bacteria, viruses, or fungi, and also water-based harmful chemicals, toxins, have a very difficult way of moving through the stratum corneum. So when we mentioned that a layer or two of stratum corneum is lost every day, we know that we have nothing to worry about because the stratum basale replaces that through its constant cell division. But what happens if pressure from the outside prevents the loss of stratum corneum? Well, the stratum basale cells don't notice much of it and they keep dividing. So if the stratum corneum is under physical pressure, such as wearing a tight shoe or toes rubbing onto each other, pressed against each other, if you're a guitar player or a violin player, the fingertips that push down on the string have their stratum corneum compressed. And in those areas, the stratum corneum doesn't leave because of the pressure, but it keeps accumulating into what we call a callus. So this is a thickened layer of stratum corneum, usually in areas that under pressure. And so this process that keratinocytes go through, they're born as a daughter cell of the stratum basale, and they leave the epidermis as stratum corneum. This is a process where the cells are becoming filled with keratin protein. The process is called keratinization, and this happens over a period of 2 to 4 weeks. So the escalator that goes from the stratum basale to the top of the stratum corneum takes about 2 to 4 weeks to make one trip. This keratinization and the division of the stratum basale cells can be stimulated by epidermal growth factor that stimulates EGF, stimulates the cell division of stratum basale cells, and it can speed up this keratinization process. And we're going to say a few words about clinical application of all this. So maybe let's start down here. We already mentioned skin grafts. So whether we are putting the person's own skin on a large injured area, whether we are growing stratum basale cells in a culture and using that to replace lost skin area, it is important that living stratum basale cells are included in there because this is giving the epidermis a chance to rebuild itself. So if in a skin graft there's only stratum corneum or lucidum or even stratum spinosum, the skin cannot regenerate and stem cells are needed there. Another clinical application here is psoriasis. In psoriasis, this escalator that goes from stratum basale cells to stratum corneum cells, the corneum layer, speeds up, and it's shortened to 7 to 10 days. An abnormal keratin is made, And the stratum corneum cells hold onto each other a lot stronger through the desmosomes. So instead of the stratum corneum coming off as small, tiny microscopic chunks, it is shed as scales, excuse me, larger sheets of the stratum corneum, and that becomes visible. Now, psoriasis and the speeding up of cell division of stratum basale or speeding of keratinization can happen for many different reasons. We're not going to go into the clinical details of psoriasis, but it's important to know that if you hear this word psoriasis, you know that this is an abnormally fast production of epidermis. And a lot of the times this has something to do with inflammation of the underlying areas. So our next large layer in the skin is dermis, but we're going to leave this to the second lecture. We're going to have outrageous entertainment and lots of interesting examples for how dermis and the accessory structures maintain homeostasis, where they come from. So I'm going to close here. Let me get out of screen sharing here. So this is the end of this recording of the first part of chapter 5. If you have any questions, please don't hesitate to ask. And again, the instruction, what this recording is replacing, should be in your class shell. So I'm going to close right here and see you guys next occasion in lab or lecture.